So now we're going to jump into what we know about the neurobiology and neurochemistry of arousal and sleep. So, below you can see a uh, mechanistic image documenting some of the mechanisms in the neural circuitry that we now know is involved in sleep. As you might expect, many different areas of the brain are involved in what seems to be a very complex neural circuit of sleep. On the bright side, you don't have to memorize this entire circuit, which remains in a discovery phase. I would not say that we completely understand the mechanism and the entire circuit of sleep. But here's what we do know. We know that there is powerful chemical control over sleep. If you don't sleep well one night, you might feel sleepy during the day and need a nap. And there must be some sort of physiological mechanism that monitors the amount of sleep that we get. The simplest explanation is that the body produces a sleep promoting substance that accumulates with wakefulness and is destroyed by sleep. The substance can't be in the general circulation of the body. The hemispheres of the brain can sleep at different times in some species, like the dolphin. And this suggests that the chemical must be produced within the brain and act locally within it. Adenosine is one of the chemicals involved, and is, as I'm drinking my espresso right here, uh, directly related to caffeine's effects on arousal. Adenosine is a nucleoside neuromodulator, a neurotransmitter, that is released by neurons engaging in high levels of metabolic activity. It may play a primary role in the initiation of sleep. And if you gunk up that system with caffeine, you go against sleep, promoting arousal. Uh, Right. So when neurons are active and need fuel, estrogens convert glycogen into glucose for them. As part of that conversion process, the astrocytes produce adenosine and pump it into the extracellular space, where it has an inhibitory effect on neural activity and is therefore sleep promoting. During slow wave sleep, neurons in the brain rest and astrocytes renew their stock of glycogen. Allele variation in the enzyme that breaks down adenosine may play a role into why some people need more slow wave sleep than others. And remember that caffeine is an adenosine receptor antagonist. So, the idea is that it would promote arousal and wakefulness by disrupting this adenosine promotion of sleep. So neither sleep nor wakefulness is a unitary condition. If you are fighting to stay awake, you will impair ability to concentrate on other things. If I scare you, you become more alert. Circuits of neurons that secrete at least 5 different neurotransmitters play a role in some aspect of an animal's level of alertness and wakefulness, what is commonly called arousal. So we're going to discuss 5 of these arousing neurotransmitters. And I want to point out that none of these has a singular function, but all are implicated, in promoting arousal. These include acetylcholine, norepinephrine, also known as noradrenaline, serotonin, histamine, and orexin, also known as hypocretin. One of the most important neurotransmitters involved in arousal is acetylcholine. 2 groups of acetylcholine neurons, one in the dorsal pons and one's located in the basal forebrain, produce activation and cortical desynchrony when they are stimulated. Acetylcholine agonists increase EEG signs of arousal, and acetylcholine antagonists decrease them. Acetylcholine levels increase during REM, but are very low during slow wave sleep. So think acetylcholine generally promotes awake and arousal. Here is just one graph illustrating how acetylcholine levels change in the cortex and hippocampus in different levels of the sleep cycle. Uh, you know, Basically, you can see that there is some uh, uh, variation across the sleep wake cycle, with increases occurring in REM sleep in both the cortex and the hippocampus relative to slow wave sleep. Investigators have known that the catecholamine agonists, such as amphetamine, produce arousal and sleeplessness. These effects appear to be mediated primarily by the noradrenergic system of the locus coeruleus, located in the dorsal pons. Locus coeruleus neurons release noradrenaline, and the locus coeruleus is a dark-colored group of noradrenergic cell bodies located in the pons near the rostral end of the floor of the fourth ventricle. It is involved in arousal and vigilance. Here you can see the mean firing rate of noradrenergic neurons during waking, slow wave sleep, REM sleep, and again when the animal wakes up. So here you can see that norepinephrine neurons fire when awake, and that stimulation of stimulation of locus coeruleus neurons using optogenetics causes immediate waking. This has been used as support for the idea that norepinephrine increases vigilance, paying attention to stimuli in the environment, etc. This is really a long-standing role for noradrenaline, and more sort of a confirmatory observation with optogenetics than anything. Serotonin is a third neurotransmitter that plays a role in activating behavior. Almost all of the brain's serotonergic neurons are found in the raphe nuclei, which are located in the medulla and pons regions of the brainstem. The raphe nuclei, a group of nuclei located in the reticular formation of the medulla pons midbrain, are situated along the midbrain. Here you can see the mean firing rate of serotonin neurons across the sleep-wake cycle. You can see they decrease over the course of the entire sleep-wake cycle. Using optogenetics, we know that stimulation of raphe nuclei can increase locomotion and arousal. This also can block the synthesis, and if you block the synthesis of serotonin using PCPA, this can reduce overall levels of cortical arousal. Again, supporting this notion that serotonin is generally arousing and activating. Now we have histamine, the fourth neurotransmitter implicated in the control of wakefulness and arousal. It is primarily released from the tuberomammillary nucleus, a nucleus in the ventral posterior hypothalamus, just rostral to the mammillary bodies. It's involved in cortical activation and behavioral arousal. Histamine causes the release of acetylcholine in the cerebral cortex, thereby increasing arousal. Its activity, the activity of histamine neurons is high during wake, but low during slow wave sleep and REM sleep. Blocking histamine receptors with antihistamines like Benadryl, the first generation antihistamines that pass the blood-brain barrier, the ones that don't promote drowsiness. The reason that they simply don't cross the blood-brain barrier to act at the centrally located histamine receptors. But anyway, if you block those, you decrease waking and increase sleep. Orexin, which is also called hypocretin, which makes a lot more sense in the context we're using the term. So, when orexin was discovered as a molecule, there was great debate in the field as to whether or not it played a role in increasing eating appetite or increasing wakefulness and arousal. I actually think there's a little more evidence to support a role of wakefulness and arousal, which would be more in line with its term hypocretin. Orexin specifically is associated with feeding behavior. That's the name origin. But regardless, hypocretin neurons are cell bodies that secrete hypocretin molecules or orexin molecules. These cell bodies are located in the lateral hypothalamus. Only 700 hypocretin neurons occur in the human brain, but they project to almost every part of it. Hypocretin has an overall excitatory effect, stimulating the release of hypocretin in the TMN, for example, activates the histaminergic neurons located there. Narcolepsy is caused by generation of these neurons. There are a lot of great studies out there done in Dobermans. Interestingly, Doberman pinschers have a greater prevalence of narcolepsy than other species of animals. So Dobermans are the most common model to study narcolepsy behavior, but you see this degeneration of hypocretin neurons and that is associated with the onset of narcolepsy. Here you can simply see firing rate of orexin neurons during exploration, active walking. You can see them slowing down during quiet walking and are very slow during slow wave and REM sleep. Sleep is controlled by three factors: homeostatic, allostatic, and circadian. If we go without sleep for a long period of time, we will eventually become sleepy, and once we sleep, we will be likely to sleep longer than usual and make up at least some of our sleep debt. This is homeostatic. We're keeping the balance. The primary homeostatic factor that controls sleep is the presence or absence of adenosine. Sleep, it's important for us to stay awake in some conditions, like when we're threatened or searching for water when dehydrated. This would be called allostatic. Allostatic refers to the reactions in the environment that override homeostatic control and is mediated by hormones and neurotransmitters. Circadian factors, like time of day, tend to restrict our period of sleep to a particular portion of the day-night cycle. Circadian control of sleep cycles is primarily mediated by the suprachiasmatic nucleus of the hypothalamus and the pineal gland melatonin. And recall that, I will most certainly ask you to describe, the neurobiological basis of circadian behavior on the exam. That's when I promise you'll get as an essay. There are 5 sets of arousal neurons that control slow wave sleep: orexin, histamine, noradrenaline, serotonin, and acetylcholine. But what controls these arousal neurons? Oh, and I'll try to provide the term. I'll accept norepinephrine, noradrenaline, and orexin or hypocretin. These are synonyms. They all mean the same thing. So what controls these arousal neurons? The answer is the ventrolateral preoptic area, a group of GABAergic neurons in the preoptic area whose activity suppresses alertness and behavioral arousal and promotes sleep. So the idea is that there's basically a flip-flop between the VL preoptic area or ventrolateral preoptic area and the activation of the brainstem arousal systems, including acetylcholine, noradrenaline, serotonin, histamine, and the one you don't see there, hypocretin. So, if one is high, it will be driving the other, and vice versa, sort of like a seesaw. And through this mechanism, by the POA activation of these arousal networks. You would see mass release throughout the brain and a wide array of network effects that can occur to promote arousal and activation. Hypocretin plays, a specific role here. It, I guess, can have specific effects on direct effects on the arousal systems that are in addition to the activation of the GABA neurons of the preoptic area. And there it's interesting to note the similarity between the location of the orexigenic neurons and those of the preoptic area. All of these are hypothalamic containing neurons. I mean, in a lot of ways, the orexigenic neurons are very similar to the preoptic area neurons. And this is just a way of adding them into the equation and the balance of that seesaw. So the overall effect here is that with prolonged wakefulness, what you're going to see is an accumulation of adenosine, which would have an effect on the preoptic area, which would then have an effect on the orexigenic neurons and on the arousal system. But you're also going to have a number of factors acting on the hypocretin orexin system as well. These include the biological time of day, hunger signals, satiety signals, drugs of abuse. I have published multiple papers on the hypocretin system involving, how it can influence and how drug use can influence the system. It does. But regardless, the point here is to show that this is complex. The mechanisms are not as simple as it would be wrong to think of this simply as the POA driving a few arousal systems that wake up the brain. That would be an unreasonable oversimplification. This is still a bit of an oversimplification, and maybe even arguably a little bit of a cop-out by basically saying, the mechanism is very complex. Here is a sort of a watered-down version of the complexity. But I think that is a fair place to end. I don't know that it is reasonable to expect you in an introductory level class to memorize 1,000 different specific mechanisms and try to put those pieces of the puzzle together to completely describe the neurobiology of wakefulness, because frankly, it's still in a discovery period. We don't have the full mechanism worked out yet, but this is generally, how it would work. Wakefulness leading to adenosine having direct effects on the ventrolateral preoptic area, which is going to interact with the arousal system. But you're also going to have interaction with the lateral hypothalamic orexinergic neurons, also known as hypocretin neurons. These are going to be acted upon by other signals. Some people think of orexin neurons as only conveying hunger signals, for example. It's more complex than that, and this is just one way of illustrating that complexity. Now here we see our bringing back our flip-flop teeter-totter analogy, where you see activation of the ventral lateral preoptic area, which down on the bottom would then turn off the locus coeruleus, the dorsal raphe, these projection arousal activating neurons, and also inhibit hypocretin neurons or orexin neurons, thereby promoting sleep. The orexin neurons would also be typically activating the locus coeruleus and dorsal raphe nucleus neurons, in addition to the dopaminergic ones, I might add. On top, the opposite occurring, a state of wakefulness is being promoted. because the ventrolateral preoptic area is being inhibited. Orexin neurons are being stimulated, as are the arousal neurons at the locus coeruleus raphe. So, think of those GABAergic neurons in the ventrolateral preoptic area as sort of arousal brakes. If you take those off, you will teeter toward the bottom, which is a state of wakefulness involving, or you will teeter to the top, which involves a state of wakefulness involving the activation of the locus coeruleus, dorsal raphe, and orexin hypocretin neurons. We know that REM sleep is controlled by a flip-flop very similar to the one that controls cycles of sleep and waking. The sleep-waking flip-flop determines when we wake and when we sleep, and once we fall asleep, the REM flip-flop controls our cycles of REM sleep and slow-wave sleep. There's also a pharmacology of arousal. We know several drugs that can promote vigilance. One is amphetamine. Amphetamine does many different things, but it enhances serotonin, noradrenaline, and dopamine transmission in the brain to promote arousal and vigilance. Amphetamine is not the drug that is given out to the soldiers and the senators and the presidents anymore. Instead, we give something called modafinil, which promotes wakefulness through similar mechanisms, but has a reduced chance of promoting drug abuse. Caffeine blocks adenosine receptors. I mean, here I am, I'm on my second espresso, double espresso of the day. Nicotine stimulates acetylcholine receptors to promote wakefulness, also improve cognitive function. You also have sleep-promoting pharmacology. Alcohol produces a biphasic effect on sleep, but at high levels induces it. Barbiturates and benzodiazepines both do. These are both really more allosteric modulators at the GABA-A receptor, as you should know at this point, well, especially benzodiazepines. Antihistamines block H1 histamine receptors. H2 histamine receptors are located more in the periphery and involved in the allergy response. Those that pass the blood-brain barrier, the first generation ones like Benadryl or diphenhydramine, induce sleep. One way to save $5, when you go to the pharmacy, is to note that almost, probably about 20% of drugs in the pharmacy are diphenhydramine, being repackaged in a variety of ways. Diphenhydramine is just your pink little pill that blocks H1 histamine receptor centrally. It is going to induce drowsiness, so it's marketed as a sleep aid. It's also what they put in, various nighttime solutions of cold medicine. You're also going to see it in your anti-allergy medications. So there are three different, rows that drug is already featured in, and it is the most common there. Interestingly, I've done some work with first-generation antihistamines. Most of these drugs are very dirty and bind to very many receptors in the body. Very few of them would probably pass FDA approval today and are sort of grandfathered in. They do activate many different receptors. And diphenhydramine's even been paired with drugs of abuse at times. One, every now and I remember there was a big supplier of heroin for a long period of time, combining heroin with diphenhydramine. So the heroin is typically brownish, combined with the pinkish diphenhydramine, made the product come out as yellow, and they called it cheese. But anyway, crazy stuff, those antihistamines. And I will stop there.